everybody. I scheduled this a little bit last minute, so I'm using not as nice a mic as I have been previously. But we're going to do a Biology Unit 2 paper today. So as usual, before we start and we properly get into it, we're going to take a look at the instructions. They are on the top of the paper there. So we have got write your centre and candidate number. You'll be given those before every single GCSE exam. There's nothing too surprising or out of the blue there. I've also said you must write your answer in the space provided. That is a common mistake that people make and one that I've been seeing a lot more frequently. So we'll go through that if it comes up at all. And it says the usual instruction, don't use gel pen. Total number of marks for this paper is 90. So I'm going to scroll up again and just confirm that this is indeed the higher tier paper. I haven't accidentally opened the foundation paper. And then finally, the last thing that I want to note is that the question 10C is a quality of written communication question, i.e. it's our essay question for this paper, which, it means that we can take a look at how to structure that type of question later on. So as usual, the main focuses that I have whenever I am doing one of these paper streams is we're going to look at our command words and how to answer our questions properly. We're going to look at how to get the right amount of information in whenever we've got, say, a 2 marker versus a 4 marker, like how much detail do we need to include. And we're going to take a look at the examiner's reports and see how other people compare doing these same types of questions. So we use the examiner's reports to see where sort of areas for improvement for most pupils are. Again, with the chemistry exam every single year, it's people needing to better understand the difference between atoms, ions, molecules, compounds, and all those very similar sounding words. As for what comes back in the biology report, that tends to be different from each year to year. So we're going to get started here, and we will start with question 1. So question 1. The diagram shows one strain of the flu virus, and it has a surface protein, as is pointed out here. And we've been asked to say, what is the term given to the proteins on the surface of a virus? So viruses have a very particular sort of structure that we know about. They've got this membrane, and then on the membrane there are our antigens. Okay, so our antigens are the things that our bodies can see and detect. So I'm going to write that in here. I'm going to write this in the answer space. So antigens. And then underneath, we've got the flu vaccine contains a weakened virus which is injected into the body. White blood cells respond to the weakened virus and produce antibodies. Explain why the viruses in the vaccine must be weakened. And that is one of the sort of questions that immediately it seems like if you know it, that it's common sense, but if you don't know it, that it might be a little bit harder. So why do we have to have a weakened version of the virus? Well, firstly, who are the main sort of people that we're going to be giving a vaccine to? So a vaccine is given to tends to be the very young, very elderly, people who are sick with other things. So they tend to be people who cannot risk getting more sick. So basically, we give a weakened version of the virus so as not to make the person receiving it sick. The thing is, that's not a lot of information. Receiving it, I can't spell, apparently. Receiving it sick. That's not a lot of information. So we're going to take a look at what is given to us in the mark scheme and how much information they actually want for us. So we've seen line by line, this will give the numbers per mark. So the first, it does not cause the disease would be 1 mark, but it still has to contain the antibodies or stimulate the white blood cells into working. So my answer here would only have one mark. So in order to get the second mark, I have to both state why it has to be weakened, but why it still has to be the virus in and of itself as well. So, so as not to make the person receiving it sick. What's that? It still gives an immune response. Okay. So my typing does not take up much space on the paper lines at all. But remember, most people's handwriting will be bigger, or so it would probably, if I was writing that out by hand, it would probably take maybe 3 or 4 lines. So, name the type of immunity produced by a flu vaccine. Well, it's acquired. So we have different types of immunity we get. So it's an acquired immunity, and it's also active. Sorry, that's my fault. I made a mistake there. It's active is what they want in the mark scheme. It's active immunity. So active immunity means that our body has created the antibodies itself using the antigens. So we'll scroll back up here, and this surface protein, the antigen, our body learns what that looks like and is able to attack it specifically. And the process of our body learning it and being able to produce its own antibodies in response makes it active. Passive immunity, on the other hand, would be if instead of receiving the virus, we just straight up receive the antibodies. This tends to be a common approach for in nature for young babies. So, for example, a mother breastfeeding a child, the child will receive antibodies from the mum, from the mother's milk, and that is passive immunity because our bodies in that scenario are not creating their own antibodies. So it's active immunity. So name the type of white blood cell that produces antibodies. So that is a lymphocyte. Lymphocyte. Have to try and remember how to spell it. So lymphocyte. So lymphocytes are so called because they're produced in the lymph. That's kind of it. So if I move over then to the draw button. So it says draw a circle, and they've handily circled this for us, around the type of antibody that is produced in response to the flu virus. So if we look here, we've got this kind of square one, a kind of triangular one, and this trapezoidal one, I guess you could describe that as. And it is going to be, if we take a look at it. So antibodies do a couple of things. They attach on to the antigen. So in order to attach on to the antigen, it has to be a similar sort of shape. So it's like the inverse of that shape, or a complementary shape. It's very similar to how enzymes work. So for that complementary shape then, that suggests to us with those triangular proteins that we need to circle this triangular antibody. So notice this antibody has 2 ends on it as well. That's beneficial for a couple of things. One is that it can attach immediately onto, it gives it 2 chances of attaching onto one antibody, and then once it has attached, not antibody, sorry, one antigen, once it has attached to one antigen, that antigen, if it hits another antigen, can clump together, and that causes large amounts of the virus to be form a giant ball that makes it easier for our immune system to attack. It's easier to hit a bigger target. So describe and explain the function of this type of antibody in the flu virus. Well, I began explaining it there. So this type of antibody attaches, I spelled body incorrectly, antibody attaches to the surface protein known as the antigen on a virus. The other end can attach on to the same antigen on another virus, causing them to stick together. And then once it's stuck together, macrophages can eat the viral clumps. So they all clump together, and that allows for our body to get the macrophages in. So macrophages are very large white blood cells. If you see the prefix macro, then macro means large or big. So a macrophage then, and anything where you see phage in biology is something eating. So phagocytosis means to break down a cell by eating it. So anything where you see phage, it means eating or cells eating each other. Cyte is anything to do with cells. So up here, like whenever we talk about lymphocytes, it's a cell from the lymph. And anything ending in "is" means the breakdown of. So lysis is breaking down a cell. Phagocytosis is is a thing breaking down a cell by eating it. Hydrolysis is breaking something down with water, and I had another example, but I've completely forgotten what it was. Anyway, not the point. So, going back over this question then, is that the end of question one? That's not the end of question one, but it's most of question one. I want to talk a little bit about our command words. So our command words here are explain. So 1A2, we have explain. So explain wants some amount of detail. So our command words are basically the verbs in our questions that tell us what to do. We have name. So name is the basically just state a thing, and it's very brief and short. No detail kind of required there. Name again. Then we've got draw. Draw feels very obvious. You're not writing words, you're drawing a thing, in this case a circle. Describe and explain then. So describe tells us that we want to outline the process, and explain then also says to us that we want to explain why that process is the way it is. Okay, so every year a different strain of the flu virus forms. The diagram shows a new strain of flu virus. Describe 2 ways the new strain differs from the original strain. So the original strain, one of the very obvious differences is going to be it has a different antigen. So it has a different antigen. And that's the obvious difference. So then our other difference, if we take a look here, is that it has fewer proteins or antigens. So up here, this virus has 10 antigens on its surface, whereas this one only has 7. So it has fewer antigens as well. Antigens. Now, the reason that having fewer antigens is beneficial for the virus is that the antigen is something that the body is capable of seeing and therefore attacking. And if it can attack it, it's like I said earlier, like whenever it clumps multiple viruses together, it's creating a bigger target. Well, having fewer antigens reduces the number of places where if the antigen hits it that it can actually latch onto. Therefore, fewer antigens also makes it harder for it to survive. So, use the diagram to explain why a new flu vaccine is given every year. The antibodies produced by the previous vaccine would be ineffective against the new strain. So the reason it's going to be ineffective against the new strain is that, remember, the antibodies that we produce are this triangle kind of shape, this inverted triangle shape, because they are attaching on to this triangular protein. So if they're attaching to this triangular protein, then this new rounder protein isn't going to be able to effectively attach onto it. So we want a new vaccine that is more likely to attach onto it. So one of the things, if you work in healthcare, you're particularly old, elderly, you're particularly young, or you have a compromised immune system, you will be offered the flu vaccine every single year, and it's in order to keep those people who are most at risk the safest. And they have to predict, there's a lot of science done in order to predict what type of strains of flu are most likely to be prominent every year. Now, sometimes they get it incorrect and people still get very sick, but they do this prediction to get ahead of any curve and protect people against future flu strains. So that's the end of question 1. So the way any GCSE paper is structured, it's structured in such a way that the easiest question should be at the start. And that wasn't too difficult a question, but it was quite a long question. It was, let's count 1, 3, 6, 7, 8, 12. So it's 12 out of the 90 marks. So that's a lot in that first question, a lot of information. So we're going to take a quick look at the examiner's report. And we want the examiner's report for the higher tier. So, is that visible on screen? It is. Okay, so the unit overview. A wide range of marks were obtained by candidates. This could indicate the paper allowed candidates different abilities to respond positively to the questions posed. Only more capable candidates were able to access the more difficult and challenging marks available. The paper contained a good number of discriminating questions to allow for a good level of discrimination. So in exam marking, what they mean by discrimination is being able to tell between candidates who really understand the content and really know how to answer the questions from people who maybe just haven't really quite grasped it and can only do the basic level of question answering based on what they've learnt. And in general, the way a paper is structured, the first questions are the easier ones that most people will be able to answer, and the later questions tend to be harder. So it's good to try and get as many marks early on as possible. So this opening question was accessible to candidates of all abilities and was generally well answered. In part A, only a minority of candidates were unable to identify antigens and lost marks in 2 for failing to explain that viruses used in vaccines need to be weakened. Most candidates were able to recognize active immunity, name lymphocytes, and identify the complementary antibody. However, part 4, some candidates incorrectly focused on what happens after clumping takes place rather than explaining how the action of the antibodies leads to clumping. Some candidates were confused with antigen-antibody complex with enzyme action and used inappropriate active site terminology. So I did say earlier that it is very similar in terms of the fact that it's both proteins involved in antigens and with enzymes, and it's also the fact that it's very shape based, so it's similar to enzymes in terms of how they work. However, they are not they are not the same, and it is important to make sure that you have clearly identified which is which whenever you're writing your answer. So you don't want to say things like active site, even though both are shaped determinate. And then as for our antigen antibody complex, I didn't use that term in my answer, probably should have. But a complex is basically in biology a number of things stuck together. So in part B, most candidates were able to correctly describe differences in two strains of virus, but a significant failed to use the diagram as prompted in the question and therefore made no reference to the shape of the antigen, no longer being complementary to the antibody. Yeah, that is kind of important thing to note, in fairness. Right, so we will continue on to question 2, and before I do so, I'm going to take a drink of my water. Right, so the diagram shows a pair of chromosomes. So we've got a T allele for a tongue ruler and a B allele for brown eyes, a small T allele for a non-ruler, and a small blue allele for blue eyes. So I can actually roll my tongue. And it's actually been disproven that tongue rolling is 100% genetic. There are some people who can learn it. And then we've got blue eyes and brown eyes. So use evidence from the diagram to explain what is meant by a gene and an allele. So a gene is the actual a gene is the actual like the placement of the genetic code that codes for a particular characteristic, and the allele is essentially the variant of it. So the allele, the gene for eye color, and the allele for brown or blue eyes. So the gene says what color eye I get, so it says what the physical characteristic is, and the allele is the variant, so whether or not it's brown or blue. And the way that they've worded it in that mark scheme is gene is part of a chromosome that codes for a characteristic, and the allele is a different form of the gene, whether or not it's dominant or recessive. Okay, so then we've got what is the genome. So the genome is basically the entirety of your genetic code. And again, I want to be particular with wording, so I'm just going to check what it says in the mark scheme. And it says all of the genetic material of an organism. So yes, the entirety of our genetic code is what I had said. So it's fairly similar meaning, but yeah, where possible, try to use C as wording because C are very particular about all that sort of stuff. So the diagram shows the range in size of the genomes of 4 groups. So bacteria, which are very small, have a very small genome. Insects have a larger genome but quite a wide range. Mammals have a large genome but a small range, and plants have a large genome and a very large range. So, which group of organisms have the smallest genome? And the answer for that is going to be bacteria. I was going to say bacteria. That's not a word. But yeah, bacteria is going to have our smallest genome because our size of the genome, if I get my highlighter here, size of the genome is shown to be this bit at the bottom. So which group has the smallest genome? It's going to be the one that is like around the smallest number, which is bacteria. So bacteria has the smallest genome. Which group of organisms has the smallest range of sizes of the genome then? So that's going to be the one that fits into the smallest amount of space, and that's going to be mammals, because we can see that mammals will always have somewhere between 9 and 10 arbitrary units, and they only take up about a third of that space, where even the bacteria take up 2/3. So the much larger potential size in genome, but a much smaller range of possibilities of size. So the smallest range is mammals, which humans are mammals. So a student examined the diagram and claimed that the larger the genome, the bigger the organism. Name a group of organisms which does not fit this trend. I feel like quite obviously plants. Like not on purpose, but have a plant with me in the office today. So this is a sage pineapple. I got it in home base, and yeah, it's quite clearly much smaller than me. I'm a mammal, but this sage pineapple might have a larger genome. I don't know. So yes, it doesn't fit that trend because some of the larger genomes could be in plants, and plants can be a range of sizes, including smaller than us. Right. So it says use evidence from the diagram, which basically just means read information from the diagram, state it, and explain how that fits your answer. Okay. So question 3 here then. Oh, wait, before I move on to question 3, let's take a look at what the examiner's report says. So, a large number of candidates lost marks in Part A because they did not use the evidence from the diagram. Textbook definitions were textbook definitions which were not evidence-based were not rewarded since this was a skills question. So if we go back up to question 2, they're saying that people didn't get this first one because they were using the definitions that they had learnt off for they had learnt off for these things, allele and gene. But it does in the question say use the evidence from the diagram. So this is going back to using our command words and making sure we are truly not just reading what the question is, but understanding what they're asking us when they're asking us a question. When they say use, they're telling us where we should be getting our information from. So if they say use your knowledge, it will be the stuff that you have learned in classrooms. If they're saying use evidence from a diagram or a graph or something, a piece of information that they've given you, take it from that piece of information. Okay. Let's see. In Part B, descriptions of a genome were often vague and failed to gain both marks. In the interpretation of the diagram, many confused the size of the genome with the range of the genome. So they're saying that people basically swapped their answers. So some people said mammals when they were supposed to say bacteria, and bacteria when they were supposed to say mammals. An encouraging number were able to select the appropriate evidence and explain it in sufficient detail and to obtain one or both marks in Part 4. So, okay. So now we move on to question 3. So, So 3A, a photograph shows a human blood smear. The cell A, or name cell A and describe its function. So cell A, there's a lot of similar looking ones, and it seems to be kind of darker around the edges. So that's a slightly thicker around the edges, meaning it's a biconcave shape because we can kind of see a little bit of light passing through it. It's thicker around the edge than it is in the middle. So that tells us that it's going to be a red blood cell because our red blood cells are circular, but they're missing a nucleus. And because they're missing their nucleus, they're sort of flat in the middle. So name the cell. So highlighting name the cell. It's going to be the red blood cell. But notably, this question is 2 marks, and that second mark comes from describing its function. And describing the function of the red blood cell was quite simply to transport oxygen around the body. Describe one way cell A is adapted to carry out its function. I mentioned earlier that it's got this biconcave shape that comes from it losing its nucleus, and it loses its nucleus in order to get it loses it in order to have more space to be able to take oxygen up in hemoglobin. So that's one way in which it is. That's one way in which it's adapted. So, Part B transports waste. Part B transports the waste product urea. So B is in this sort of blank space here of the blood smear. So name Part B and give one other waste product it transports. So Part B is plasma. So we've got four main components of human blood. We have platelets, we have plasma, we have red blood cells, and we have white blood cells. In question one, we kind of talked about what white blood cells because it's all to do with immunity. Red blood cells we've just talked about. Platelets are used then to clot blood whenever someone has been injured or gets a cut or anything like that. And then plasma is used to dissolve things and carry them around the body. So there are various different things that we want to get rid of in the body. And my brain is failing to think of absolutely any of them. I can think of all sorts of different things, like different types of hormones and stuff gets carried around in the blood. What would it be? Concave nucleus contains hemoglobin, plasma, carbon dioxide. Okay. Yes, plasma does carry around carbon dioxide, and for some reason my body, not my body, my brain refused to remember that it was carbon dioxide because I kept thinking, oh, carbon dioxide attaches to red blood cells. No, it doesn't. It attaches to, or not attaches to, it gets dissolved in plasma. Okay, so photograph 2 shows us blood clotting. Name the part of the blood which is needed for blood clotting. Well, I mentioned earlier the four constituents of human blood being red blood cells to carry oxygen, white blood cells for immunity, plasma for dissolving things and transporting things in, and then platelets, which are then used for blood clotting. So platelets. Use the photograph and your knowledge to describe the process of blood clotting. So again, it's probably really important here to look at the use of the word use the photograph given previous things that we've discussed as part of the examiner's report. So when someone gets a cut, platelets begin to clump together in order to initially try to seal that cut, and they will get red blood cells to attach onto that. And then there's a whole process to do with fibrogen and all of that sort of stuff. So the way I really hoped they'd have it in a little bit more detail than that, but they didn't. They've literally just listed words. So fibrogen. So the platelets of fibrogen, and whenever we have a cut, that fibrogen gets converted into fibrin. So fibrin is sticky and begins to catch the red blood cells that are moving past and sticks to more of itself as well and forms that kind of mesh which traps more and more red blood cells until we end up with a scab or a clot. Okay, and then suggest why blood clotting is necessary. Well, the reason blood clotting is necessary is that our bodies have a certain amount of blood that they need in order to perform the functions around the body, namely delivering oxygen to every single cell in the body. So if we don't have enough blood, there isn't enough volume to carry all of those red blood cells, or there's not even enough red blood cells in order to get oxygen to every aspect or to every single cell in the body. So if we don't have enough blood, we die. from oxygen starvation, effectively, we bleed out. So blood clotting is necessary so as not to use blood and ensure that all of the cells in the body get oxygen. So that's the end of question 3, which is another really short question. So we'll see what is said in the examiner's report. So part A, part 1 and 2 were answered very well. Most could name and give the function of the red blood cell. In part 3, a disappointing number of candidates failed to read the question carefully, and we're not giving credit for naming urea as a waste product since it was given in the stem of the question. Yeah, that's fair enough. They gave urea in the question, so you couldn't get it as an answer because it says, urea as a waste product, name one more. So then the process and function of blood clotting was well known, though some candidates were unable to accurately name fibrinogen and correctly describe its conversion to fibrin. Yeah, they're two very similar sounding words. It's very easy to get them mixed up between each other. I will say that if you see gen usually means become. So if you think of the word hydrogen from chemistry, hydrogen means to create water. Hydrogen is not water, but it is used to make water. Fibrinogen, then, you can use that same thing of gen means to become something else. Fibrinogen will become fibrin. So we start with fibrinogen and it turns into fibrin, if that helps you remember the order. Okay, so we're going to move on. So, question 4a. Estrogen is a female sex hormone. Name the organ which produces estrogen. That's the ovary. That's pretty much it. There's not really much else to say. Then name one other female sex hormone, and it is progesterone. So if anyone is on, say, the contraceptive pill, they might take it for its contraceptive purposes or for another medical reason, such as heavy periods or something like that. Then some people will be on an estrogen only pill, or sorry, some people will be on a progesterone only pill, and some people will be on what's called a combined pill, which contains both estrogen and progesterone. So progesterone is the other female sex hormone. Okay, so the graph shows changes in estrogen level. The graph shows changes in estrogen levels of 2 females over 12 months. So basically both have these zigzag lines. They both go up and down, up and down, up and down, up and down. And this is our second person here who's just got relatively low levels. So most people do actually have, there's quite a wide range of amounts of hormones in people's blood. It does vary from it does vary from person to person a lot. So this could be normal, this could be abnormal. We really don't know. But they do follow the consistent pattern of dropping, rising, dropping, rising, dropping, rising, and they'll drop and rise to similar levels each time. And you also notice that these are like relatively well spaced. So some of them might be shorter or longer than others, but they are relatively consistent. So give the range in estrogen level in female A and female B in arbitrary units. So the range, so we want to look at the lowest level for female A, which appears to be about 200. I don't see anything less than 200. It would be easy then to, so I do a dot right about there-ish. It would be easy then to leave that there and then read off 400 as the top. However, we can see that there's this reading over here, which is a bit higher and probably reads about 540. So I would put female A's estrogen level as ranging from 200 to 540 arbitrary units. Female B, on the other hand, very low levels would be a bit harder to read, but seems to be nothing below, I think, that reading there, which is 40. And I see Nothing above this reading over here at the 7.5 month mark, which is at about, that is 140 as well. So that ranges from 40 to 140 for female B. So suggest why female B might have difficulty becoming pregnant. Use evidence from the graph to support your answer. So it says use evidence from the graph, which means we want to, where is the, yeah. So use evidence from the graph. So that means we want to be looking at this and stating information from the graph to tell us how to answer a question. So female B has consistently low levels of estrogen compared to female B, and this means that it could affect her fertility. And if that's the case, then female fertility is the ability to get pregnant. So estrogen is low, unable to release eggs, or unable to ovulate. Okay, so suggest a treatment which could be used to help female B become pregnant. It would literally just be shots of estrogen. So for the body to get more estrogen. That is a genuine fertility treatment where people do get, they take injections of estrogen on a regular basis in order to encourage their bodies to ovulate. So before we move on to question 5, doing the usual changeover and going to looking at what the examiner's report has to say about question 4. In part A, most candidates were able to correctly identify the ovaries as the site of estrogen production, though a large number were unable to correctly name progesterone as another female sex hormone. Spellings such as proestrogen, which looks like a mixture of estrogen and progesterone, were not rewarded. So they wanted the correct spelling of progesterone. So possibly important to learn how to spell that. In terms of like learning to spell things correctly for GCSE Biology, genuinely recommend getting a group of friends together, picking a list of keywords, getting a list of keywords together, and playing hangman with each other, because you will probably remember it after a while, and hangman can be quite fun, especially if you're doing it with friends. So, in part B, a disappointing number of candidates were unable to correctly read the values from the graph, though most went on to suggest why low estrogen would cause difficulty becoming pregnant and how fertility could be treated. So most people got the second part correct. So moving on to question 5. Describe how water is lost from leaves in transpiration. So I have my plant again. So this is a pineapple sage plant, and you can see that the leaves here are green. And I cannot get a microscope and get a cross section of this area, but if I could, I would see various structures within the leaf itself, including the likes of the stomata, which whenever it's open is a potential source of water loss. So whenever the let's see what it says in the mark scheme. So the mark scheme says, so evaporation. So evaporation is the first part of transpiration, where again, these stomata, whenever they're open, they lead into basically it's an opening into spongy mesophyll, where water can be lost from, and then that is done through the process of diffusion. So diffusion is things moving from a high gradient to a low gradient in order to eventually have no overall gradient. So they would take any 3 from that. So if I were answering it, would probably mention evaporation from the spongy mesophyll through the stomata. Whenever you are doing leaf cuttings, so if you were to get your own plants, what they actually do is they cut some of the leaves in half so as to limit the amount of transpiration and therefore water loss, in hopes that the plant retains enough water so it can re-establish roots and therefore grow from a cutting. Okay, so the photograph shows the lower epidermis of a leaf. basically, normally when we look at our diagrams of leaves, what we see is a cross-sectional area. So they're cutting through it and we're seeing that slice in half. This photograph, on the other hand, is actually not of a leaf cut in half and looking at the top and the bottom, but rather just looking at the bottom of it and looking what how the bottom of it looks. So cell X is it's a guard cell because and it's important to know that it is a guard cell and not a stomata. So the stomata are the holes or the spaces that lead to the spongy mesophyll, but the guard cell is the cell that surrounds it and opens and closes the stomata. Kind of like how your pupil is a hole in your eye and your iris controls how much or how little light gets in by opening and closing um, that sphincter. Okay, and that is the end of question 5. Question 5 is incredibly short, it's only 4 marks. Okay, so question 5. So, some parts of this question proved challenging for a significant number of candidates. Many candidates were unable to give a description of transpiration. However, some mixed up the sequence of evaporation and diffusion and unfortunately lost 1 mark. So, in the mark scheme, they said give any 3. So, the important thing for them is not necessarily mentioning all of the steps, but rather that they're mentioned in the correct order. So, therefore, it's useful then to realize the order in which those things happen. So if you say diffusion before you say evaporation, that's incorrect. We don't want to do that. You want to say it's evaporation. Okay. Right. So basically, you could have as much detail as you want, but if you say something incorrect, they can't give you full marks because it's incorrect biology. You have to have in a short question, it's sometimes better to know that you've hit all the marks and not add any more detail that could potentially be incorrect than it would be to write as many things down as possible and write something incorrect. Write all of the things you definitively know are correct, and if you think you're short, if you don't think your answer is long enough, then maybe add something you're less sure about, because they don't take away marks per se. It's just you don't want to write anything blatantly incorrect because that will be marked. That can't get you full marks. Whereas having enough information but it not be overly detailed, but you have enough to get the points, will get you full marks. So sometimes less is more. It's really a judgment call for yourself whether or not that works for you and whether or not you will actually benefit from that sort of approach. But some people will have a much harder time if they write things they're unsure of compared to if they write three really good points that they know are definitely correct compared to 4 or 5 points because they're not sure about the 4th or 5th point, but surely more information is better. Not always. Write enough to get your marks and don't write anything you're unsure of if you think you have enough detail. If you don't think you have enough detail, it's sort of the risk is kind of balanced, but that actual interpretation will be up to you whenever you sit your own exam. It's going to be a judgment call. Right, so back to the question. Or not question, back to the paper. Oh, there's part C of 5. Whoopsies. Oh well, we'll do part C of 5 now. The number of stomata on the upper and lower epidermis of a leaf can affect the rate of transpiration. The table shows the distribution of stomata on the upper and lower epidermis of 4 different plant species. So, upper epidermis of A is 60, lower is 94, upper 360 versus 0 on the lower, 136 on the upper for plant C and 369 on the lower for plant C. And then for plant D, we've got 124 on the upper epidermis and 254 on the lower epidermis. So, species A grows in hot countries with hot climates. Explain why Species A is best adapted for the best adapted of the 4 species to grow in countries with hot climates. Use the data from your table to support your answer. So this is important, again, where it says use data from the table, it means we have to use information that we were given in the question in the table. So, species A would be the best acclimatized because it has the fewest stomata overall. So basically, the warmer it is, the more evaporation is possible. But in addition to that, so it has the fewest overall, and it also has the fewest on the upper epidermis, because as heat rises, potentially water could get caught in the lower epidermis and not evaporate off. Depending on wind, you can still get more evaporation, but on that upper epidermis will result in a lot of water loss. So, suggest which one of these plant species grows on the surface of ponds and lakes. And that one's actually fairly obvious, and the answer is B, because there has to be a reason why there's nothing on the lower epidermis when normally the lower epidermis has more. And the reason for that is the plant would just drown. The plant would drown if it had lots and lots of stomata on its lower epidermis and it was on a lake. So let's take a look at that. So part C1, frequently losing marks for failing to use data and for incorrectly referring to plant species A as having the least number of stomata on both upper and lower epidermis. So it has the least overall, but it doesn't have the least on the lower epidermis. Species B does. The majority of candidates gained at least 1 mark for appreciating the need to conserve water in hot climates. In contrast, part 2 was well done, with many candidates being able to select the appropriate species and explain the adaptations. Okay. Here we go. So, meiosis. So meiosis is a type of cell division. We have 2 main types of cell division that we care about in GCSE Biology Unit 2, meiosis and mitosis. So, sorry, I've been talking a while, so I really need to drink water. So, diagrams A, B, C, and D show a cell dividing. Give the letter of the diagram which represents meiosis. So the answer to that is D, and then give 2 pieces of, I'm sorry, meiosis is not D, it's because you get 4 daughter cells, don't you? It splits and then it splits again. Yeah, okay. So it's C. Because it has 2 divisions and we get 4 cells at the end. For some reason, I got myself confused and was thinking, oh no, it only has it splits into 2, but no, it splits into 4. So in this first cell division, so it splits twice. In this first cell division, there's this you've got the full 46 chromosomes in both of these cells. It's only in that second division does it break down into our haploid cells. So a haploid cell contains half of the amount of genetic code necessary, and then that joins together with another gamete to form a diploid cell, which has 46 total chromosomes. So where does meiosis take place in males? And the answer, it's in the testes. So the testes are used to produce sperm cells, and sperm cells are the gametes in males, whereas ova, or an ovum, singular, ova, plural, are produced in ovaries in females. So it's the testes where the sperm is produced. So testes is, I just think this is really interesting that the root word for testes comes from the same root as testament, and it just means to bear witness, which is it's an odd word. So gametes produced by meiosis show genetic variation. Name the process in meiosis. which causes genetic variation. So that is independent assortment. And basically what that means is whenever all of the cells are lining up in the middle of cell division, that they sort of swap about what's on their genetic code. So not every chromosome is a copy of the chromosome that it came from. So some will have genetic information from parent 1 and some will have genetic information from parent 2. So the table shows the number of chromosomes found in the body cells of 3 different species. So a worm has 12, a goat has 60, and a human has 46. How many chromosomes are found in the gametes of a worm? And the answer is going to be 6, because you will always have half of the amount of genetic information in the gamete. So 12 divided by 2 is 6. So cells can also divide by mitosis. So mitosis is how every other cell in the body divides except for ova and sperm. So give 2 functions of mitosis in a living organisms. So in a living organism, mitosis has basically only got 2 functions, and that is for growth, so to make cells make more of the cells so that it can provide provide for a larger area. For example, the size of my skin as a grown 27-year-old is going to be very different from the amount of skin required for a 7-year-old. So therefore, I need more cells in order to cover myself. So my body created new ones through the process of mitosis. And then the other one is for repair. So repair then is if you have an infection and that causes some of the cells in that area to die, that your body can create new ones. So for growth and repair are the 2 functions of mitosis in living organisms. Okay, so that's the end of question 6. Let's go to the mark scheme, and we've got most candidates were able to identify the diagram representing meiosis, but marks were often lost for giving the theoretical descriptions of meiosis rather than using evidence from the diagram. So again, this is using what we physically see in the paper rather than things that we inherently know. A lot of the things in more recent GCSE papers have been about interpreting data and interpreting data correctly. This is particularly important for your Unit 3. I know that this is a Unit 2 paper, but in your Unit 3 paper, you will see lots of examples of this. So let's see. While most were able to name the testes as the site of meiosis in males, some contradicted themselves by quoting testes/sperm. So they wanted the site, they didn't want the gamete. So testes is the site, sperm is the gamete. Only a minority of candidates were able to correctly identify independent assortment as the source of genetic variation. So chances are this statement here, whenever teachers read through an examiner's report, they will usually take information from it that will state, oh, this wasn't done well, or this was done well, and will change their notes accordingly. So you might have learned since then that independent assortment is that swapping of genetic information that ensures variation between individuals. It means that I'm not a carbon copy of my sister who, she didn't get the same set of genes from my dad as I did. She didn't get the same set of genes from my mum as I did. She got a different set because that independent assortment means that it mix and matches what kind of alleles are on each pair of chromosomes. So, part B then was generally well answered, although part 3, the usual growth and repair, were often embellished, and quite a few candidates gave replace old cells and repair damaged cells, which were the same marking point and worth just 1 mark. So yeah, it is just growth and repair. So I explained them and embellished them and explained why they're the answer. But remember that whenever I'm doing these sorts of past paper question explainer videos, they are explainers. They're explaining why we are they're explaining why we are answering the questions the way we are. I'm not inherently just giving you what the answers are. I'm also explaining to you why the answers are the way they are through explaining the structures of the question, um, and also like why, how you can think about the answer and how you can arrive at that answer by yourself. So, question 7. So, question 7, we've got these two images of blood vessels. So, blood vessel A, which has a very wide lumen, and blood vessel B, which has a much smaller lumen and a much thicker muscle around the wall. So, name each type of blood vessel, and before I do that, I'm going to take another drink. Because I've been talking for an hour now. So, vessel A is a vein, vessel B is an artery. They both have quite wide lumens, so they're both for transporting large amounts of blood, whereas a capillary will be very, very small. And I know that vessel B then is my artery rather than my vein because it has that muscle around it, which allows for us to withstand the high pressures that are in arteries. Right. So describe two differences visible in the diagram between the vessels. So they have obviously got a different width of lumen, and they've got one has more of that one has more of that sort of like muscular structure, which my brain is refusing, uh, absolutely refusing to name what it is. So thin wall or thick wall. A has large lumen, B has small lumen. Yeah, it doesn't actually tell me that. I know somewhere in the back of my brain I know the word for the stuff that makes up the wall, but it is refusing to come to me right now, so I'm just going to continue on with the question. So the graph shows the rate of blood flow in arteries, capillaries, and veins. So in arteries, we have this really high intensity blood flow. That's in part because the arteries are coming from the heart, so they get the most amount of pressure applied to them. In capillaries, the blood flow slows all the way down because it's a very thin area and not much blood can pass through. So to compensate for that, we just have loads and loads of arteries. And they are that thin because they are required to be in order to squeeze out the gases in order for gas exchange to happen. So that's getting carbon dioxide out in the lungs and O2 in, and for every other cell in the body, it's for getting oxygen into the cells and CO2 out. So, describe and explain the change in the rate of blood flow between arteries and capillaries. So, in between arteries and capillaries, the rate of blood flow decreases drastically. So, that's describing it. So, describing it is stating specifically what the change is. So, we go from a high rate in the arteries to a low rate in the capillaries. And then explaining it would be stating why, and it's that the arteries have a wide lumen capable of facilitating lots of blood flow, whereas the capillaries are much tighter and much smaller, making it harder for blood to pass through by design. And then explain the change in the rate of blood flow, explain how this change in the rate of blood flow helps capillaries carry out their function. So I said that it was by design. So capillaries are for the purpose of gas exchange. This means that narrow width allows only one red blood cell to pass through at a time, which allows for that gas exchange to happen. So that's your explanation. And specifically in the mark scheme, they're saying that it allows more time for diffusion. Okay. So question 7. Surprisingly, the photographs were not well known by candidates. The vein was often named as a capillary and the artery identified as a vein. So whenever I said they both have wide lumens, that means that those two, that they're going to be those major blood vessel. So it's important to know that a wide lumen, even though the artery's lumen was smaller, means that they're going to be veins or arteries and not capillaries. And that kind of muscular wall is characteristic of arteries. Again, not a thing in capillaries. So capillaries they're only one cell wide. It means you're only two cells away from a cell getting oxygen. And it's important that they are very small and very narrow. So that is like an important thing to know about the cross-sectional area of it's an important thing to know about the cross-sectional area of the diagrams. So it's worth looking at those photographs and memorizing them and being able to know the difference. So then underneath we've got some candidates did not gain credit as they did not use any comparative terms to describe the differences in the two vessels. Another common error was to describe the artery as having a thicker cell wall. It's not a cell wall because the wall itself is the cell. The artery is not a cell, it is a vessel. It is made up of more cells. So yeah, that is indeed an error. As for not using comparative terms basically means you have to compare A to B. So saying that A has a thinner wall and B has a thicker wall, that is a comparative term because you have to state the difference between one and another. You can't just say thick wall. You have to say which one has a thick wall, or A having a larger lumen and B having a smaller lumen. You have to state which one, and that is your comparative term. So your comparative term is, this one is like this, and you don't necessarily have to say that the other one is like that, but it's obviously better. So it's better to say A has a thin wall whereas B has a thick wall, and A has a large lumen whereas B has a small lumen, than it is to just say larger or small lumen. It is important that we are using our comparative terms. All right, so I didn't finish that. So most were able to use the graph to describe the changes in blood flow, but not all were able to suggest the explanation. And then many candidates correctly described the role of the capillary in the exchange, but only more able candidates appreciated the reduced rate of blood flow would allow for more time. Okay, so moving on to question 8. I can maybe breathe for a second. Right, so how many questions are in this paper? I probably should have checked before I started. 10. Is it 10? Yeah, it's 10. And 10C is the last question, which is the essay question. So back to question 8 then. So flower color in pea plants shows variation. Explain what is meant by the term of variation. So variation is basically different specimens of the same species will have different characteristics, and this is useful for usually for things like survival. So differences between individuals of the same population. Okay, so flowers of pea plants can be purple or white. Name the type of variation shown by color in pea plants, and that is a phenotype. So a phenotype is like a physical trait that you can see the difference between. Okay. Other examples of variation in pea plants include the length of the pea pod, whether the peas are smooth or wrinkled, the number of peas per pod, the number of root nodules per plant, and whether the plants are tall or dwarf. Give the letters of the two examples which show the same type of variation as flower color. So this is going to be, again, like a sort of a physical characteristic. So it would be B for whether the peas are smooth or wrinkled, and also for E, which is whether they are tall or dwarf. I, coincidentally, also from my plants, actually have pea seeds. Weird coincidence. That's not, I didn't actually plan for that. So, the root nodules of pea plants contain nitrogen-fixing bacteria. You might remember this from Unit 1. The table shows how the average number of root nodules per plant varied when different masses of nitrate were added to the soil in each plant. So 0 added, 24 nodules, 50 milligrams added, 19 nodules. So we can see as we add more nitrates to the soil, the number of nodules decreases. Calculate the percentage change in the average number of root nodules per plant when pea plants were sown with soil containing 200 milligrams nitrogen compared to those with none. So, this is my drawing tool. So, we look at the difference. So we want to calculate the percentage change. So we do 24 take away, it's 207 over 24. And that has to all be times 100. So I'm going to get a calculator up because I don't actually know that off the top of my head. You won't be able to see the calculator, but just trust me, I am typing it in. So 24 minus 7 gives me 17. Divided by 24 is 0.07 times 8, and then times by 100 gives me 70.8. So my answer is going to be 70.8%. Okay? Oh, I didn't actually write the 100 times 100. So 100 turns it into a percentage. So the way we calculate the percentage change is by getting the difference, and it is the difference over the original number. Okay? So the original number minus the new number, all divided by the original number. So that's 17 divided by 24. And then we have to times it by 100 to give us an actual percentage number. So it's 70.8%. Okay, so what evidence suggests that the variation in the average number of root nodules per plant is caused by the environment? So the evidence that we have is essentially that this is quite consistent, that the more nitrates we add to the soil, the fewer and fewer nodules that the plant has. Now it still needs nodules because it needs the nodules in order to take up some of those nitrates, but it will make fewer and fewer and fewer the easier and easier it is to get nitrates from the soil in order for the plant to make proteins. So basically the trend is quite consistent in that as we add more nitrates, the fewer root nodules there are. Okay, so 8 in the examiner's report. So part A, the variation was very poorly described, with few appreciating that variation represents differences within the same species of a population. In part 2, many were confused not only by types of variation as continuous or discontinuous, but as the type of variation with the possibilities of causes of variation, whether or not it's genetic or environmental. So, continuous or non-continuous is basically, so if something is continuous, it can be a range of things. Factors like height are continuous because you can be 140 centimeters tall, you can be 180 centimeters tall, you can be 160, you can be anywhere in between any of those values. You can be greater than 180 centimeters tall, you can be shorter than 140 centimeters tall. That is continuous because it could be any hypothetical value between those points, and in addition to that, it can change over time. Although that is a genetic factor, although there are some environmental inputs. Let's not get into that. That's a whole rigmarole of things. So that is what a continuous variable is, or a continuous variant in the human genome or human population is. And then a discontinuous variable is like yes or no. I can or I cannot roll my tongue. I have brown. Well, no, that's a bad one. I have 5 fingers on each hand, or I don't. That's discontinuous, where it can be yes or no, or only a discrete number of possibilities, whereas continuous could be anything. Okay. Anything else? Despite this, most were able to identify the examples. In part B1, very few candidates achieved all 4 marks in this calculation, as they did not correctly identify the change was a minus decrease and seemed unfamiliar with the concept of 2 significant figures. Oh, right. So, was that a mistake I made? Let's see, go up here. It does say to 2 significant figures. I made that mistake as well, so I'm going to correct that. The answer here is actually going to be, I'm going to change the color to show that I made a mistake, is 71%. Where is that? And then the other thing here with the percentage change is that they seem to be very particular about the fact that it is a negative change. I don't like that. But essentially they're saying it should be minus 71%. So yeah, the actual calculation is still correct, but it should be it should be 7 minus 24, not 24 minus 7, because it's a decrease. So it's the new value minus the original value divided by the original value times 100, which would get you minus 71% to 2 significant figures. So 2 significant figures basically means that that it being to 2 significant figures basically means that there are two numbers that can convey information. And you might think, oh, but 70.8, surely the 0 and the 70 doesn't convey information. No, it does, because it tells us that it's not 69% and that it's not 71. It tells us that it is at 70. So that 0 does have significance, but in this case it rounds up, so it'd be minus 71%. That is my mistake. I will admit to that straight out. Yeah. So, what evidence suggests that variation in the average of average number of root nodules vary? Okay, but anyway, question 9. Okay. A breed of dog. In a breed of dog, the allele for a black coat is big B dominant to the allele for a brown coat, small b. A dog breeder wanted to find out the genotype for a black dog. He crossed his black dog with a brown dog. Describe the type of describe the type of cross the dog breeder carried out. That's weird wording. Okay, so it's a test cross, which basically means it's being used to figure out what the original type was. So it's a test cross. I need to go back to blue. Okay, so it's a test cross. People do this with plants all the time. So complete the Punnett squares for all of the possible crosses. So the brown dog is 2 small b's. I really thought that would have been the other way around. I thought they would have asked us for the brown dog's alleles, but they didn't. So so here it will be B, 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 B. Actually, well, all of them are going to be. So it's big B, small. So it's big, small, big, small. And this one's going to be small, small. And I made a mistake in this one. That is going to be small, small. So then for this type of gamete, we're going to have the 2 big B's there. And in this one, all of our all of the genotypes we end up with are going to be dominant. So if the dog had a heterozygous genotype, which basically means that the two letters are different, then it's got a 50% chance of its offspring being black and 50% chance of its offspring being brown. Whereas if it is a homozygous gamete, then both of the letters are the same, and all of the offspring have that heterozygous heterozygous set of genes. So the big B and the small b, meaning that they all come out with the dominant gene, which is the black dog. So explain how the breeder may be able to tell the genotype of the black dog from the color of the pups. Well, basically, you would expect 50% of the pups for the 50% of the pups for the heterozygous alleles to have a brown fur and 50 to have black fur, but you would expect 100% of them to have black fur if it was a homozygous allele. So homo means same, hetero means different or across. Okay, so go to question 9, and so the vast majority of candidates answered this question well. While a disappointing number were unable to name a test or a back cross, almost all candidates were able to complete the Punnett squares correctly and use these to explain how the genotype of the black dog could be identified. Some candidates lost marks for the misuse of the terms heterozygous and homozygous. So heterozygous and homozygous are for like the actual offspring. So yeah. Right, so on to the final question. So yeah, if they're homozygous, they're the two B's, and if they're heterozygous, they're different. So anyway, question 10. Deoxyribonucleic acid, don't know why that was so hard to say, or DNA, codes for proteins in cells. Describe the structure of DNA. So this is a thing you're sort of expected to know, and the main things are it has a phosphate backbone. It contains a phosphate backbone. It contains a ribose sugar, and it contains a nucleic acid, which will be A, G, C, or T. They are together. Two pairs of amino acids will be across from each other. These will be either AT or C and G due to their length, and will be A, G, or C and T, but they will form like this ladder shape that will twist, and this twisting is this twist is called the double helix. So in the mark scheme, they want 3 notes from, it's a double helix structure. It contains a sugar. That sugar is called ribose. It has a phosphate backbone, and it's got the base pairing. So that's the nucleic acids A, G, C, and T, with A and T pairing together and C and G pairing together. So describe how DNA differs from one person to another, making them unique. Basically, the sequence of the sequence of nucleic acids differs from person to person and will be in different amounts from person to person, and this allows for like different amounts of proteins to be coded for. Okay. So then we've got diagram A shows a section of DNA strand from normal skin. Diagram B shows a section from a cancerous skin cell. The table shows the amino acids coded for by the different triplets. Describe and explain one environmental factor which may cause skin cancer. Describe how this factor affects DNA and what effect this has on the functioning of DNA. Okay, so this is actually quite a nice essay question because they've given us a fairly consistent, like they've given us points for our structure. So we've got describe and explain. Do they have this separated out first? No, they haven't. Okay, sometimes they do. Sometimes they'll have like a bullet point, do a bit under it, bullet point, do a bit under it. Okay, so one factor which may cause skin cancer. The most obvious factor is obviously how much sunlight or how much radiation or UV light a person is exposed to. So some people, you might have seen, there are pictures of lorry drivers, and one side of their face will be very aged and old looking, and it will be because that's had so much sun damage, whereas the side that's not on the side, the half that's not on the driver's side, will be much more young looking, and it's because of that damage that is caused by constantly being hit by light from the window versus the side that's on the passenger side, which will be in shade. It is actually genuinely worth looking and Googling that image. So basically, the amount of sunlight, the amount of UV radiation, etc., that a person is exposed to can affect their risk for skin cancer. The reason for that is that the energy from the radiation can damage the sequence of DNA, changing different letters. So in this case, from diagrams A and B, it changed a G to a C. So instead of coding for serine, then leucine, then valine, then proline, it doesn't, it stops coding for the leucine and then codes for double the amount of valine, which means that you're not getting the same amino acids that you need in order to you're not getting the same amino acids from your DNA that you need for your body to function properly, and then that can cause some of your cells to essentially change how they're structured and do things out of control. So cancer cells tend to grow at very fast rates, have uneven borders, not really stick to the same type of tissue that they're supposed to, not differentiate correctly compared to other cells, etc. So you can draw that kind of thing in your answer, but because it says give evidence from the diagrams, use it to support your answer. So basically, that radiation that you're exposed to through sunlight can cause a change in one of the bases in a base triplet, which changes the amino acid that it's coded for, and therefore change change what proteins the body can make, and then that can lead to cancer. So the way, so they've got in their indicative content for this answer, they've got the UV radiation leads to a mutation. So the G is changed to a C, valine instead of leucine. DNA changes the DNA code. The final protein is different or cannot function. So to get full marks in this, they wanted at least 5 of those 6 points. I don't think I used the word mutation, but I think I said everything else. And they want good spelling and grammar for it. So they want you to be able to structure it. And whenever you're writing it out, just constantly refer back to the previous page where it has that bullet point structure listed out for you that can help you. And that's really it. And I think that brings us to the end of that paper. I hope that was helpful. And if it wasn't helpful, if you could leave a comment and just tell me what you thought, where there could be improvements, if there's anything that confuses you. Also, at the minute, there is a link in my YouTube bio to a Google's form question. So if you have any particular past paper questions that you want me to do, anything you're confused about ahead of your exam, let me know. The cutoff date for submitting questions is Thursday the 9th at midnight, and I will be answering those questions on Saturday morning. I don't know what time yet, but that is the plan. Tomorrow evening, I'm going to be doing another live stream. This time it's going to be of a chemistry unit 2 paper, so if you want any help with that, you're more than welcome. So that will be on the 6th of May at 7:30. Anyway, that is me done for this evening. Hope that was helpful, and I will